any of this would be possible without actually the key help of or two people we have today with us. Is Mo from University of Ottawa as well. Dr. Mohamed Al-Khalaf has been actually a really champion of trying to bring community together within Canada and also around the world. And Hallie Arnott from the University of Ottawa, and she's the president of the CBS Ottawa Student Chapter, which is the Canadian Biomaterials Society. My name is Emilio. I'm at University of Ottawa Hard Institute, and I work in biomaterials and also smart material for tissue engineering. And to me, it's truly a privilege organizing this. And this is not only our seminar, if everyone's in here, okay? So I'm gonna do the first slide, which is a little bit of housekeeping to try to keep us all in, in time. The first one that is used to remind you guys that the seminar is gonna be 30 to 40 minutes interrupted, and it's gonna be followed up by 10 minutes of question and answers. Today we have Hallie that is gonna be the moderator for the questions. And at all times, try to keep, you have to keep actually your microphones and your camera off, because otherwise you're gonna have popping things back and forth. And if you have issues with the audio, you can actually connect by phone. And we have a toll-free number, and this is the meeting code. You have it in there. So the only thing you need to do is, dial the number and enter the code. And if you are in the US, you have to do exactly the same day with that's the phone number for the US, but the code is exactly the same. At the end, if you want to actually ask a question, you have to send a direct chat to Hallie, and Hallie is gonna actually ask the question on your behalf. And it will be nice if together with your question, you add from which institution you're coming from. That's nice, because in that way, we're gonna have a little bit of scope from where are we at the different places. And we are recording this, and we're gonna make it available in our YouTube channels, and when it's available, when I actually put the Twitter, the link in either BEaTS Research or Mo's account. And hopefully this is gonna be the first of many. So now I'm gonna actually pass the control to Hallie, and Hallie is gonna actually introduce us.
Hi, everyone. Thanks for joining us today. My name is Hallie Arnott, and I'm a graduate student at the University of Ottawa, studying in biomaterials at the Variola Laboratory. Today, we have the pleasure of speaking with Dr. May Griffith about regrowing human corneas as an alternative to donor transplantation. And next week, we'll be talking to Dr. Hamer about lanthanide-based nanomaterials for biomedical uses beyond imaging. So to get going with our presentation today, Dr. Griffith is a world-renowned scientist in the field of regenerative medicine. She has developed the first biosynthetic corneal implant that has demonstrated effectiveness to promote endogenous regeneration of diseased human corneas. Dr. Griffith leads a dynamic team now based in Montreal that seeks the translational development of novel technologies for corneal repair beyond pre-made implants. Dr. Griffith has also been an advocate to mobilize her implants and technologies to low-income countries. She has mentored and supervised many students who have also become passionate about regenerative medicine. Dr. Griffith holds a Canada Research Chair Tier 1 in Biomaterials and Stem Cells in Ophthalmology, a Caroline Duran Foundation Chair as well. And she is also the director of the cornea and anterior segment at the FRQS Vision Health Research Network. So thank you very much, Dr. Griffith, for joining us here today.
Just a recap for anybody who's listening who's not familiar with regenerative medicine. This is basically an area where we combine biology, physics, chemistry, engineering, and of course medicine to replace and regrow failing or failed organs. And as you can see in this slide, pretty much every organ in the body has one or more groups working on trying to replace it and regenerate it. So stem cells are, of course, the fundamental resource in regenerative medicine. And very quickly, stem cells are just undifferentiated cells that grow into other types of cells in the body. They're embryonic stem cells and adult stem cells. And now we have pluripotent or reprogrammed stem cells. And you can see that's a world of possibilities where you can get stem cells to differentiate into neuronal cells that might be brain cells, lungs, heart, liver, kidney. There are a whole bunch of possibilities. The commonality is that in most stem cell therapy, you take out the stem cells from the body, you expand these in a dish. For example, blood cells with a genetic disorder, you can reprogram them to make them healthy again. You can use induce pluripotent cells. But the main thing is expansion. You're expanding this outside, you're manipulating maybe, and then we transplant it back into the patient. What we're doing is something that is not quite the same. This is how people in the eye world or in the cornea world are doing the stem cell expansions and transplanting it back into the cornea. It works really well, as you can see. This is a cornea that is very, very highly compromised. You can see it's vascularized, it's cloudy, it doesn't look anything like a cornea, and this does not look anything like an eye. But growing corneal epithelial cells in a dish and transplanting it back into a patient, you can see that it's cleared up that entire cornea. Now, this is great. There are a couple of problems. One is with this epithelial therapy, it basically repairs the epithelium. If the layers underneath are compromised, you still need a transplant. And also we need a GMP lab. So this is good manufacturing practice, and we need a very controlled lab with very highly skilled people to basically grow up these cells in a manner that is safe for transplantation. So as Hallie mentioned in the introduction, I do a lot of work that's targeted towards the developing world, where GMP labs may be a luxury that they cannot afford. We also look at the delivery of adult mesenchymal cells, and this is some work that we've been doing with folks in Ottawa, mostly Dr. David Kortman and his group. And there is also stem cell therapy for animals. So for sports animals, they're starting to grow tendons. For companion animals, there's joints. There's also MSCs. And cell therapy is big in the veterinary world as well. What we're doing is something that's a little bit different, and it's more suitable, I think, for countries which cannot afford to have GMP labs at all the hospitals or clinics. And that's to try and regrow the organs of patients from the inside. And this is using biomaterials. So basically what we're doing is using biomimetic materials that we implant into a patient that will stimulate the patient's own cells to grow and differentiate into the organ. So what we're doing now is working with a couple of different tissues and organs, but our main one is the cornea. And this is because the cornea is a very simple tissue. It's got three main layers: an epithelium, a stroma, and an endothelium. Most of the stroma is made of collagen, and there's some proteoglycans. It's also transparent, which means for research, I can actually see what's happening. I can see cells growing in. I can see vessels, which are not desirable, but they do grow in. And you can also track your drugs or your bioactives. If you label them and you make them fluorescent, we can track these with laser confocal or in vivo confocal microscopy. So corneal blindness, why are we interested in this? It's something that affects people of all ages. It could be unilateral in one eye, so as in this boy, or bilateral as in this baby. And it's not something that's in the developing world. We can see people in the developed world, mostly children who have issues. In the developing world, it may be a problem with vitamin A deficiency. in the developed world, it's unfortunately usually some infection. And the most common infection is herpes simplex virus 1. I mean, we're all dealing with viruses right now with COVID-19. Herpes simplex is another virus. It's another envelope virus, and it tends to affect children. And it is a very problematic disease. It causes inflammation, keratitis, and it causes a person to become at high risk for rejecting a cornea. So I'll get to that later. Now, the other thing is that 53% of the world's population have no access to cornea transplantation. Cornea transplantation is what was used now to replace any of these damaged corneas. Corneal blindness also affects animals. And this is a horse with a very bad corneal infection. They are susceptible to injuries, ulcers, and here's a dog with an ulcer, here's a dog with another ulcer. So I'm going to talk about human patients, but also where possible, I'm going to talk about what we're doing with pet animals, or what we hope to do with pet animals as well. So we work with biomaterials. There are different types of biomaterials. There are polymers. Polymers are just long chains of materials. They can be natural, such as collagen, proteoglycans, cellulose, starch. So these latter two are from plants, and these former two are found in animals. There are synthetic polymers as well, polyurethane, which range from floor covers to the plastic on the soles of your shoes, polyvinyl alcohol, silicone. So there are different types of materials, and then there are hybrids between natural and synthetic polymers. Now, we tend to work mostly with natural polymers because we're trying to get the body to regenerate and to try to coax cells to differentiate. It's really difficult to use synthetic polymers because they tend to be at best inert. So we tend to work with natural polymers. But those of you who have worked with collagen would know that collagen is very highly degradable. It's soft, and often we get it from animal sources, which nowadays we know it's not such a good thing because there's zoonotic viruses and other pathogens. So prions, viruses, bacteria, they are very much transmissible to humans if we're not very careful. So we work with mimics of natural polymers. We've been working with recombinant collagens, and we've now been working with collagen mimetic peptides. So basically, we design them. We can get Emilio's lab to make them, which he has been doing. They have a synthesizer. But for clinical trials, we will get companies that are certified GMP, or basically that means that they can make clinical grade materials. So there are synthetic proteoglycans, there are also other bioactive peptides and synthetic lipids. So we've chosen natural materials, as I mentioned. And we work on the cornea, but we've also been collaborating with Emilio on skin. And this is also from the Heart Institute with Emilio and Eric Surinen using collagen to try and make repairs to the heart. So what are we doing now if somebody has a damaged or diseased cornea? Well, the only widespread mode of treatment is with donor transplantation. And here's a donor cornea. It works very well in most cases, except for the high-risk cases. The big problem, of course, is worldwide shortage. So only 1 in every 70 patients is getting transplanted. And there are 12.7 million people on waiting lists. So these people are waiting a long time. In Canada, in some provinces, there are 2,000 to 3,000 people altogether, and they're waiting for up to 2 to 3 years. These are artificial corneas that are in clinical use, but as you can see, they don't look very much like a human cornea. We wanted to make something that is very natural looking. And so I'm going to tell you about what we have done. This is our implant. It is basically a very clear material that looks like a thick contact lens, and we can implant it into a patient and affect regeneration. So basically we are able to make something that looks like a natural cornea. So there are very many different steps, and we'll go through some of these from all the way from designing the new materials to designing how these implants would go in, whether we would use molds, 3D printing, and then of course there's a lot of checking to do. So we do most of our work in vitro in the lab, in culture dishes, and only very few ever go into animals. That's because of the 3 R's. So basically it's refinement, reduction, and so what we do is just test only 1 or 2 models in small animals, and if those work, then we test them in large animals before going into clinical trials. So designing an implant, we use our knowledge in biochemistry, biology, and basically engineering. I've had many people work with me in the past, and with some of these former graduate students, postdocs, colleagues around the world, we've been developing different materials and developing methods to put them together, and this is the end result, a cornea. So again, these are more implants. They're made of collagen silk, and this is what they look like. As I mentioned, just like thick contact lenses. They're not very special looking, but I can tell you that it is quite special to try to get these things to be transparent. It's actually pretty darn difficult. We're using peptides now, so we can make peptides, and we get these peptides together with synthetic materials. It could be PEG or HEMA. These are conjugated. We're now trying to make longer peptides that do not need to be conjugated with synthetic materials. As well, we're trying to hybridize materials with silk to basically get our implants. So as I mentioned, we do most of our in vitro testing in the lab. We look at exposures to different chemicals that we use for cross-linkers. We look at different peptides and see how they affect cells. So for example, here we have no peptides, cells are clumped. If we put just simple little peptides, which are 4 amino acids, so going from a fibronectin-based peptide, RGDS, which is very common, you get cells to spread out. But if the cells see this DGA motif, which is more of a collagen-based peptide, it thinks, okay, I'm on collagen and I should migrate. And here you can see the filopodia. So peptides are actually a very strong platform to try and develop materials. And of course, we test a lot of these materials in organ culture in the lab. So this was actually done by Christopher McTiernan in Emilio's lab. And these are some students and our surgeon, Dr. Robert, who are testing materials in our lab in Montreal. After all the in vitro testing, we then go to animals. We have different animal models ranging from mice, guinea pigs, rabbits to a couple of different strains of mini pigs. So we use Yucatans, and we also use Gottingen pigs. This is a very large New Zealand white rabbit, which was used when I was in Sweden. And this is Per Fagerholm, the Swedish surgeon I've been working with. And here you can see we've been testing allografts, comparing it with human recombinant collagen and human recombinant collagen with MPC. This is a phosphorylcholine, which is basically an inflammation suppressant. So we suppress unwanted blood vascular vasculature. And we basically do in vivo confocal microscopy, as well as other checks, which we do in a clinic for human patients. So this is in vivo confocal microscopy, and you can see when we do our 12-month mini pigs, we can check all the different layers of the corneas. The other thing about in vivo confocal microscopy is that you can check the animals without needing to sacrifice the animals. And with the pigs, which are so expensive, we cannot afford to have pigs that we sacrifice every few months. So what we do is we give them an eye exam instead every few months. And we keep these animals for the whole course of a year. What you can see here are epithelial cells, which are basically they form a little cobblestone pattern. The nerves underneath, the stroma, and the endothelium. For human use, we do everything in a clean room. Here you can see two people basically gowned up in the spacesuits and a respirator. And that's what we're using to do the manufacturing. These are some of the biospherics units. And this was our first clinical trial with human recombinant collagen. And this is the surgery, which was done by Dr. Per Fagerholm in Sweden. So for those of you who are not aware of corneal transplantation, you can see that it is not a very delicate surgery. You might think because it's the eye, the surgeon has to be really careful, very cautious. You're going to see that it's actually quite robust tissue, and the surgeon is not being very delicate at all. So he's using a knife basically to cut into the cornea. And what he's doing is really hacking at the tissue to remove it. So this is after he's removed the tissue. Now he's going to take one of the biosynthetic implants, and these ones are made from recombinant human collagen type 3, cross-linked with carbodiamide. So this is just EDC NHS chemistry. And what Per is doing is just cutting out a piece. So he's going to cut out a piece, and he's using a circular knife or trephine to do this. So next, the piece is going to be picked up, and you can see this is the piece. It's strong enough to be manipulated. And now he's, whoops, I think we hit a boo-boo there. So he's going to put this into a patient. So now he's tapped it into the patient's eye. And he's going to be suturing these in. Yeah, so you can see that the sutures are going to go into the sides of the edges where the implant is. So he's actually not suturing into the implant. He's going to be making a mattress suture right here. So we now have 2, 4, and 8-year results from these patients. We're still finishing up our 8-year manuscript, but the take-home message is we have not had a single rejection. We've had patients who've been regrafted because of disease progression or astigmatism or problems with the contact lens. So because these were the first implants, surgeon was actually pulling on the sutures a bit too tightly, that he made some dents into the surface of the cornea. So these implants were not the strongest as well. So that was something that we were also trying to improve. But we have not had a single rejection, and we're really pleased with that. You can see that the corneas have regrown their tissue, because when we've come in to the patient, they were cell-free. And at 24 months, you can see the epithelium compared to a human donor cornea and a normal healthy cornea. The nerves are there, stroma, and we didn't touch the endothelium, it's still intact. What you should be noticing is the difference in the nerves between the biosynthetic and the human donor cornea. And here you can see these nerves are not in parallel bundles because it takes a lot longer for the nerves to sort of normalize in donor allograft. And you also see these other cells. These are dendritic cells or antigen presenting cells. So these are immune cells. We don't see them in the biosynthetic because we don't have allogeneic cells, and we don't see them in the normal cornea. There are very few of these, and they're usually immature or tolerogenic. But we do see them in the allograft. In our patients, we had a 10% rejection rate, which is the average for human donor corneas. So once we've done that with patients who have keratoconus or central scarring, we wanted to move on to these very sick eyes. And that was the aim, because these are the patients who can't even benefit from donor corneas if they were very readily available. So we did a graft on these patients after we've tested them in a variety of animals. We've tested them in mice, rabbits, and 2 different strains of mini pigs, and this is the final human patients. So we had them in 3 different groups. We had patients who were just grafted to stop pain. We did not expect them to see again because these are patients whose optic nerves are shot, like from glaucoma, but they had a lot of pain. And you can see this person, his eye is just red, just completely red, which is highly inflamed. After grafting, the eye is not red. It's not a very pretty graft, but you can see that the hole is gone. And that was the only aim in this case, to not have these holes, but to make sure that the surface was rebuilt so that the pain goes away. And yes, the pain went away at 2 weeks post-operation, and at 24 months post-op, the patients are still pain-free. With burns and infections, you can see these horrible looking scars and ulcers, or here the you can't really see the ulcer, but the epithelium is peeled off with a thermal burn. And after surgery, there is recovery, but in these patients, there was a migration of conjunctiva onto the area where stem cells were missing. And so the visual recovery was not as good as we hoped. With the viral infections like herpes and fungal infection, these patients actually did very well and they recovered significantly. So we did the second group of high-risk patients at LV Prasad in India and the Filatov Institute in Europe because they had the patient population. And you can see, unlike the surgery that I showed you that was done in Sweden, and unfortunately we still do that in Canada, like the manual surgery, at the WHO hospital at LV Prasad, they use lasers. So they use a laser to cut the implant, they use a laser to cut out the damaged cornea. Here you can see the damaged cornea. This is a big scar. And this blocked vision. So the patient was 2800, so basically legally blind because of this big white scar. And here you can see calcification and erosion of the surface. So after the implantation, at 18 months post-op, this patient was 20/50. It's not perfect. Perfect is 20/20, but 20/50 is normal vision, which means you can drive with glasses. And this is just OCT showing the scarring. We did not go full depth or did not do a DELC because we were not sure what these patients would be like. So basically, only 350 microns out of the 500 microns was removed and replaced. And you can see where it's replaced, it stays transparent. This is a add-on clinical trial with the same materials that went into the human patients, but these are vet patients. So these vet patients receive the same implants, HCMPC. This is a French bulldog. with ulcer, and this is 9 months post-op. Unfortunately, this dog, I think, did well because the doggy parents never brought the dog back, and the vet said that's a good sign because probably there were no more problems. These were two dog patients whose dog parents were a little more compliant, and they brought the dogs back for the checkups for a good 2 years. And you can see here at 2 years, both dogs have clear eyes, whereas before the 2 years, they started with these non-healing ulcers. So we were very happy with that result. But the question is, do we need solid corneal implants? If we look at the third world and people who are displaced, like in refugee camps, we're not sure that they have the luxury and of an operating theater. So this was something that I was discussing with Bruce Allen, who's a cornea surgeon in the UK, as he was driving or zooming along in his little sports car in the UK, and we decided that maybe we should do something that can really get to the millions of people. Now, if you think about it, the main thing that gets to millions of people have been vaccines. And basically each person gets an injection. So we thought, okay, could we do something that's similar? If you have a hole in your tooth, you go to the dentist fills your tooth. And with these two ideas, we sort of said, okay, why don't we fill a hole like a dentist does and deliver it to people through a syringe, just like a vaccine. And this is Chris. He was at University of Montreal. Now he's at in Ottawa at the Heart Institute, and these are some bursting pressures that he did. So he was the postdoc that worked on the liquid cornea, and he found that he could make an implant where he was getting good bursting pressures. So this is at least 7 times higher than the highest intraocular pressure recorded for people. We did a lot of in vitro characterization, and we finally put these implants into pigs. So Chris went to Sweden with his liquid cornea, and Alex and Per, the surgeons in Sweden, put it into a pig. These are in vivo confocal observations showing that at 12 months, the corneal epithelium had regenerated, so did the nerves, and so did the stroma. Fiona Simpson is a PhD student in my lab. She and Chris worked on the project together. Fiona did the cell work, and she also did work with dendritic cells to make sure that the materials were immune compatible. The dendritic cells are the main cells that would present antigens to the immune system. So we want to make sure that dendritic cells are not activated. If they stayed immature and happy, that was good. Same with the mononuclear cells. We don't want them developing into pro-inflammatory macrophages. So Fiona and Chris tested the materials and made sure that they did not activate dendritic cells compared to a positive control, which is LPS, lipopolysaccharide. And Chris looked at the differentiation of macrophages and found that they expressed anti-inflammatory markers as opposed to pro-inflammatory, so basically the M2 phenotype. So we also work with other surgeons, and this is Shameen, who is a cornea surgeon at the University of Sydney. He's an Aussie, same as Bruce. Bruce is in the UK, but he's an Aussie. So we've been working with these two Aussies. And Shameen has a rabbit perforation model. So he made a 3 millimeter diameter wound, which tapered down to 1 millimeter, and he filled it. And this is 28 days, showing that the filling was stable. And we did the pigs. So these are some of the pigs that Chris did, showing that at 12 months, the corneas were stable and most of them were fairly haze-free. We had a boo-boo. In other words, we had an unexpected puncture. So there was some hazing, but despite this, the cornea was able to regenerate. So that means in humans, even if you do have a haze, it can be corrected later. You can still save the eye. This is clinical evaluation that was done. So a lot of this will be published hopefully soon. And what we're doing now is to look at some of the immunohistochemistry. And this is Fiona's work, showing that all the implants, the liquid cornea, was comparable to allografts. These are syngeneic because the pigs are genetically homogeneous and compared to the unoperated corneas. So same with nerves, we're showing that the nerves grow back by 12 months. And right now what we're trying to do is to try and evaluate different delivery systems. This is how we've been doing it before. This is one system that we have tried to evaluate. We're not sure it's going to be the best one, so we're still looking at syringes, and hopefully if somebody who's watching my talk has some ideas for syringes, we certainly welcome those. And there are other features. So with the Melio, we were putting in silver nanoparticles to try and confer antimicrobial properties to prevent biofilm formation, and that has been fairly successful with some of the implants. So here is with particles, without particles, we have biofilm, and you can see that it's fairly successful in blocking materials. Sorry for the dogs. We also tried to confer antiviral activities, and this is some unpublished work. So I guess we will be talking with Hallie and Mo about what goes online and what doesn't go online, because this is not published yet. This is just to show rabbits where there are uninfected controls and the 6-month post-op materials. So basically we are able to regenerate the corneas, although in some of these animals, what we did was we made a second infection just during these surgeries, and you can see that although they have been regenerated, the corneas were not always as pretty as the normal. So this is something that we're working on. And basically, to summarize, biomaterials that mimic the extracellular matrix, basically collagen, can be used to get implants that induce stem cells to grow or regrow. We have picked a human cornea because there's a real medical need. We can add components that control inflammation and prevent rejection. And in the future, it may be possible to avoid transplantation by repairing corneas. So that's it. Thank you very much. Oh, this is the new building that we're at. And as you can see, it doesn't quite look like a typical research lab. And that's because it's a repurposed convent. Here you can see a statue of the Virgin Mary, and this is the inside of my lab. So thank you very much, and I'll take questions.
Perfect. Thank you very much, Dr. Griffith, for an amazing presentation this morning. Can you hear me?
Yeah.
Okay,
Thanks.
great. We are now going to look at some questions that were sent to me during your presentation. So first, we'll start with a question from Alexander Steves from the University of Ottawa. He's doing his PhD in biomaterials. His question is, have you excised long-term biosynthetic grafts and tested the comparative long-term mechanical properties?
Okay, so long-term, the longest term is 12 months. And yes, we've taken these out of pigs and we've taken these out of rabbits. and we have compared the mechanical properties. They are fairly equivalent to the normal corneas, and it's not the materials, because at 12 months, the corneas have regenerated. So what we are actually comparing mechanical properties of are the neocorneas. These are the regenerated corneas from rabbits and from pigs. So what's happening, as I tried to show you, is that cells are growing in all this time, and they not just repopulate the cornea, but they produce the extracellular matrix. So at the end of 12 months, if you compare extracellular matrices, which we do in all our studies, we can show that even if we have no collagen to begin with in our biosynthetic implants, especially the peptide-based implants, at the end of 12 months, we have type 1 collagen and we have type 5 collagen. We probably have type 3 and all the other collagens there. And they're in fairly comparable quantities to the normal cornea. So that is what is most likely giving all the mechanical characteristics of the corneas at 12 months.
Okay.
In patients, obviously, we are not able to take anything out from anybody. Nobody's going to let us retrieve their corneas. But at 8 years, the patients are quite happy. They're seeing, we figure the corneas are strong enough.
Right. He had a second part to his question. Next, he asked, comparatively, how well does a biosynthetic cornea react to elevated intraocular pressure?
Well, with the pressures, they do well. As Chris tested, they are up to about 7 times higher than the normal IOP. They would not be as strong as a normal cornea to begin with, but after the remodeling and the regeneration, we believe that by 12 months, as indicated by the results from the pigs, they would be exactly the same because they have the same mechanical properties.
Okay. Thank you. So I have a question now from Dr. Al-Khalaf at the University of Ottawa Heart Institute. His question is, does this type of synthetic implant require a different type of post-op regimen, more or less immune system suppression, maintenance, lifespan of the implant?
Okay, so we're basically using the same steroids that we would use for a regular corneal graft. The biggest difference is we can stop the steroids much earlier. So with a full thickness PRK, which is the most common implant, that allografting method, the steroids are normally continued for up to 12 months. So we can stop our steroids at about 6 to 8 weeks. That's the main difference. We don't have to give as many prophylactics because they are cell-free. And being cell-free, we don't worry so much about allogeneic cells promoting immune, chronic immune rejection or stimulating the immune cells to remain inflamed.
Okay, perfect, thank you. There's another question from Alexander Steves at the University of Ottawa. He asked, how uniform is the thickness of the new endothelial layer?
That question I cannot answer because we have not been measuring the thickness of the endothelium.
Okay.
So in the animals where we do in vivo confocal microscopy, we believe the endothelial layer, well, which we don't touch, remains about equivalent. So they would be the last 25 or so microns as we go down. Mm-hmm. Now, we haven't actually looked at the thickness of each layer in the patients. What we've done is just done pachymetry. I believe that the data would be available, But anybody wanting to look at that data would have to write to the surgeons to get access. But all the OCT data should be there.
Okay, perfect. And a final part of his question here is, lastly, could this contribute to backscatter differences in the visual spectrum?
Again, this I don't know because the patients were not measured for backscatter. In the animals where we actually took out the corneas and measured transmission backscatter, we did not get anything that was very different from the normal control. So what we did from the pigs was we took normal contralateral cornea and the implanted regenerated cornea. They were equivalent. With the patients, I don't know. Because I don't think they quite measure the backscatter. What they do is they measure visual acuity and haze. And this was, depending on the patient, some patients had haze because of the sutures, and this tended to go away over time.
Okay. I now have our next question. This question is from Dr. Diego Montavani from the University of Laval. His question is, can you comment on permeability? Is this property important as a barrier in modulating exchanges with the environment?
Yes, we believe permeability is important. So with the implants we've made, we've actually tested them for permeability. And so these implants, the permeability was tested by Dr. Mitch Watsky in the US, and the latest set of implants were tested by a group in Finland, Dr. Heidi Scottman's group. And what we are finding is that the permeabilities are equivalent to the normal cornea. So it's basically the amount of exchange was measured using dextran and albumin, and they were, permeable enough to allow these materials through. We did not do anything more extensive than that. It's just dextran and albumin with Dr. Watsky.
Okay. And as a final question here, I wanted to ask, so for this type of implant, do you see maybe it becoming 3D printed in the future, the corneal implant?
There's certainly groups that are doing 3D printing. Apart from Emilio Alacon's group, Shea Conon in the UK has been 3D printing corneas. I'm sure that there will be 3D printed corneas, and more reports of those. We mold our corneas. So we've got a bunch of molds, and we find that molding is just as fast, maybe even faster, because what we do is we put the liquid between the two pieces of plastic and we press, and that's it.
Right.
As opposed to additive manufacturing, where it would take an hour to print
a little longer.
cornea.
Right.
Yeah.
Okay.
But I'm sure people, since people have started 3D printing corneas, that there'll be more 3D printed implants around.
Okay, perfect. We do have one question that came from Alex one more time. So here he's asking, is the chemical resistance comparable? Could you design increased resistance with biosynthetic cornea? Or could you design increased resistance with this biosynthetic cornea that you're fabricating?
Chemical resistance? So against chemical splashes, I'm assuming?
I would assume, yes.
Yeah. So with the chemical resistance, it's going to be a little bit difficult to do so because the epithelial cells are growing on top of it. Right. So the implants are underneath the layer of cells. So the cells are not going to be chemically resistant. So for an artificial cornea to be chemically resistant, then you're going to need to move to a prosthesis. This would be like a completely synthetic material. And there are prostheses that are designed that are completely synthetic, and they are designed to fit in to a patient's cornea, like from PHEMA. And yes, those would be more resistant, but they would not have the cell layer on top. So in what we're doing, we're encouraging regrowth of the cells, and we're encouraging remodeling. So for our system, it would not be feasible or practical, but with prosthesis, for sure, that would be something that can be done.
Okay, perfect. Well, I think that wraps up our seminar for today. Thank you very much, Dr. Griffith, for joining us here on our special seminar series with BEaTS Research Radio.
Well, thank you very much for the invitation, and thanks to everybody who's actually joined us. So thanks again.
Great. And I'd like to remind everyone who's listening that we have a seminar next week at the same time, lanthanide-based nanomaterials for biomedical uses beyond imaging with Dr. Eva Hemmer. So thank you very much, and we'll see you all next week.